Hello everyone, and welcome back to my lecture series on molecular docking. And in this lecture series I am going to demonstrate you how do you perform molecular docking using autodock. So first let us revise as what we have done in our previous few sessions. So just to revise, in very first few video I have demonstrated you on how do we create a compound library. In my previous lecture we have discussed how do you obtain and prepare the target protein. In our case we have used this cyclooxygenase 2 as our target protein, and I have also introduced you with the concept of system validation. How do we perform system validation, what are the steps that are involved in performing molecular docking and virtual screening. We have used Autodock as our molecular docking software, and Python prescription was used as a interface for running this Autodock in background. So this is what we have done. So today we are going to discuss on interaction analysis. How do we perform interaction analysis of the protein ligand complexes that we might have obtained in our previous step of molecular docking and virtual screening. So this is what the major focus of today's screencast is, as to how do we perform interaction analysis and how do we obtain the uh, complexes for protein ligand systems that we might have ended up generating during the process of molecular docking. Okay, so let us begin our demonstration. First, let me recall, or first let me show you as to what is that we have obtained. Okay, in our previous lecture, we have obtained the results in form of two files, right? We have saved a comma separated file that can be opened into any spreadsheet program like MS Excel, and in this comma separated value or in this Excel sheet you can record what are the ligands that we have used and what is their corresponding binding free energy values in kilocalorie per mole. That is what we have done. Okay, so this is one part of result that you might be interested in, and another part of result that we have done is that we have exported the entire session into an archive file. Okay, so this is what it is. Okay, and you can right click it and uh, extract it. So this can be done if you have already installed any extraction software like WinZip, or 7-zip is also one of the very popular archival software that is normally used to extract and unzip the archives file that are popularly present or that are popularly distributed on internet. Okay, 7z you can use this software. If you have not downloaded it, you can download it, and you can use that open access software, that is free software, for archiving or unzipping any type of file that is present. Okay, that is very handy software, right? So after installing you would get it something like this 7-zip, right? And you can use this 7-zip to extract any of the archives as you wish. Okay, so in my PC the default extractor is WinRAR. Okay, if you don't have WinRAR, you can download and install this 7-zip as well. That is also equally good. Okay, so once you extract your result into say some folder, it might look something like this. All you have to do is to extract everything. Okay, and you might end up with these three folder: macromolecules, ligand, and etc, etc. So your main results that you are interested in lies in this macromolecule folder. So in this macromolecule folder there would be folder for your target. So if you have kind of multiple target, there would be multiple folders over here, but since we have used a single target in our previous study, there is a single folder over here. So you can open it, and the results, or you can say the docking result for each and every ligand is represented in its own separate folder, or say subfolder, right? So there would be a folder for each and every ligand that you have docked. Okay, well, say for example, there would be a separate folder for this 2' hydroxyflavone, and all the results are saved in a separate folder. So the results for molecular docking, are in form of two files. One, what we call it as docking parameter file, and another file is in form of docking log file. It is also called as DLG file. Okay, so if you check out the properties of this file, you can see the extension over here, .dlg. DLG stand for docking log file. So this is a standard output if you are using Autodock program. Okay, now if you are using Autodock Vina, the output would be in form of PDBQT file itself. Okay, so this is the difference between Autodock and Autodock Vina. Autodock Vina would output the result in form of PDBQT file, while your Autodock would output the results in this docking log file. Okay, so we would be using this docking log file in a while. Okay, so now this docking log file has been automatically detected by my PC that it can be opened in this Vega ZZ software. Okay, so since I have installed this Vega ZZ software on my PC, and it has detected that okay, this file belongs to this software. Okay, now in your PC, it is entirely possible that this icon might not be the same as it appears over here. This file might look like this. Okay, this docking parameter file. Okay, so don't worry if icons are different. Okay, just always make sure that you are working with the right file. When I say that we want to use the docking log file, it should not happen that you end up loading this docking parameter file. Okay, so this docking log file is the file that we are interested in, right? Okay, so that is a standard docking output file that we are going to use today. That is where our discussion begins. Okay, so now I will tell you what is the exact issue with obtaining the complex, or what are the difficulties that you might face when you want to generate that COX and flavonoid complex now, right? So for doing this, for generating the complexes, you would require this Python prescription. Of course, you can open the Python prescription using the desktop icon if you have already installed it. Okay, there is one more thing that I want to share with you people, is that in our last lecture I have shown you how do we perform molecular docking, right? And then we have closed it, right? So the thing is that other softwares, when you close them, they usually delete all the content that was generated in your previous run. But that is not the case with this Python prescription, right? So if you close this software, whatever you have done is still lingering around in this software. So every time you start out, it is your job to do all the cleanup stuff. Okay, so I would recommend you that you have you develop this habit of either cleaning this software before you start the run or after you start the docking run. Okay, so in this case we are cleaning everything after we have performed molecular docking. So just check out every time that if there is anything that has been left from your previous run or from your previous analysis, just make sure that and then begin your new analysis. Otherwise you would get confused as to what you are dealing with. Okay, so now our first job would be to clean up everything that we have done in our previous docking run. As you can see in these tables, the ligands are still there that we have used in our previous run. Okay, you have got the same target over here, you have got docking results as they are over here. So before you start out anything new, it is necessary that you clean up everything that has been left around, right? So cleanup is quite very easy. Go to each and every tab. Okay, then you might want to select everything. Okay, click on the first row, then scroll down, hit the shift button and click on the last row. Everything is selected. Okay, here is the delete icon. Okay, use this delete icon to delete whatever rows that are present in your tables, right? So similarly do the same for targets. Select it, delete it, go in next tab, docking result, select everything, make sure everything is selected and then delete it. Okay, so we have deleted everything. I mean you have cleaned up everything from this table tab. Make sure you do the same in document tab. You don't have any document. Are there any 2D plots available? Nothing. Since we have not loaded anything, this 3D scene is also empty. Then you might want to uh, clean up this autodoc tab, because there would be still some files present over there from your previous run. Okay, so I would similarly select it, select everything. After selecting, it becomes blue. That is an indication that everything has been selected. All you have to do is after selection, right click and then use this delete button. It would ask you if you want to delete the folder. Yes. Okay, and do the same, right click and use this delete. Okay, it would ask you if you are sure if you want to delete it. Yes, I am very sure. Go ahead for deleting. Then it would show you some warning. All you have to do is to hit this reset logs button. Okay, so now everything is clear. You have got a new slate on which you can begin your new work. So I advise you that make sure that everything is deleted, and before beginning your new task, always make sure that you begin with a new slate as such. Okay, so don't keep anything from your previous run. That would only create confusion. Okay, so for the sake of simplicity, I would now close it and again I would open the software. Okay, so now since we have reloaded our software, let us check out if things are still there. Now you can see there is nothing. Okay, and we have a clean slate now. So now we are ready to do our new job. Okay, so as I have been mentioning you that the output of the docking is usually saved in the form of this docking log file. Okay, this docking log file, this is what I am talking about. Okay, and this is the output that we would be needing, or this is the output that we require for our subsequent process. Okay, so we would require the docking log file from each and every folder that that stores the that stores the output, right? So from each and every folder we would have to copy this docking log file into a new folder. So in this way we would need to copy a docking log file into a new folder, right? So this is what we are going to do. We are going to use this is what we are doing, right? We are on step 3, obtaining the complexes and interaction analysis. Okay, so the very first task that we need to do is to separate all docking log file. So I would rename it as 1 and I will name them as docking log files, DLGs. Right, so in this folder we would save all those docking log files. So this thing can be done in either of the two ways. Okay, you can go into each and every folder, select this docking log file, copy it, go to the new folder and paste it, right? So if you have got limited number of compounds, then of course you can do it 12 or 13 times. But what if you are dealing with we'll say some 150 or even 500 compound, then every time copying and pasting would take a lot of time, right? So the alternative option is that what you can do is that you can go to the result folder where all the subfolders are present. All you have to do is to search for star dot dlg. So star means everything that ends with dlg, every file that ends with dlg you have searched into that folder, and you might end up with all dlg files over here, right? So all you have to do is to use this control A button. Control A would select all, right click, copy all dlg files, okay, and paste it over here. It would ask if you want to replace the file. Right. So here we have the data for all of our DLGs. Okay, so in this way we have collected all the docking log files in a single folder. It would become convenient for us, right? Okay, so now since now you have all the docking results in a single folder, well, now there is a button over here by which you can load this docking log file and see how results are saved. So that is exactly what we are going to do. We are going to load this docking log file. So if you hit this button, push this button, it would ask you where your docking files are located. Go to desktop, then we have got this docking lecture, and in this step 3 obtaining complexes we have this one DLGs, right? And we would load one of the docking log file over here. Let us load the very first one, the docking result for 2' hydroxyflavone, right? We would open it. Now you can see that all the 10 poses that this software has calculated are shown over here. This is first pose whose binding free energy is minus 7.57. Then you have got second pose, you might have noted that something has changed, right? The confirmation, or you can say the confirmation of this particular fragment has changed, right? This is second pose whose binding free energy is minus 7.21. Then you have got third pose whose binding free energy is minus 7.82. Then you have got fourth pose, fifth pose, sixth pose, seventh pose, eighth pose, ninth pose, and 10th pose, right? So in this way these are 10 poses of this 2' hydroxyflavone that this software has detected. Okay, it would eventually mean that this is the orientation in which our ligand might orient in your protein. So these are the 10 best possible poses it has saved. Okay, so now the thing is that when you save the result, only the poses of the ligand are saved, right? Do you see any receptor protein present in or around here? No. So here lies the problem. What we have is the poses of these ligand that has been predicted, but while saving the result, we don't I mean the software don't save the protein molecule, right? So in this docking log file, in docking result, all you have are the different confirmation of different poses that can be attained in the protein's active site, but the protein structure is not saved over here. So our main objective now is to generate the protein and ligand complex, so that eventually later on we can analyze whatever interactions that might be happening in our protein ligand complex. Okay, so this would be our first task today. Okay, to generate the protein and ligand complex. Okay, and then subsequently we would see what are the interactions that are happening in that protein ligand complex, or in that protein flavonoid complex in our case. Okay, so in order to obtain the complex, what we would do is that we need to first uh, export all of these poses out of this software first, right? So that would be our first task. Well, we are visualizing these poses that are present in your docking log file. So first thing that we need to do is that we need to take out these poses in a separate file, right? And this thing can be done using this particular button, save as SDF, right? So you can use this particular button, or use this particular option for exporting all of these 10 poses into a separate file as an SDF file. That is what we are going to do, right? So we would use this save as SDF button, hit that button. Okay, and maybe in our obtaining the complexes and interaction analysis, we would create a new folder. We would rename it as 2 poses. Okay, we would save all the poses over here, right? And we would open it, right? And we would give it some name. What name should we give? We would give as 2 prime hydroxy flavor. And since all the 10 poses would be saved over here, we would add or append the name as underscore all poses, since all the 10 poses would be saved in this SDF file. The resulting file would be a structured data format file, SDF file. Okay, and when I click OK, or when I click save, all these 10 poses would be saved or would be extracted into this SDF file. That is what we are going to do. Save it. Okay, so let me see, in our previous folder we have generated this new folder over here, 2 poses, and in this we have got this SDF file. Okay, so let me tell you. Okay, so this is what it is. So all of these 10 poses, first pose, second pose, third pose, fourth pose, fifth pose, sixth pose, seventh pose, eighth pose, ninth pose, and 10th pose, all of these 10 poses are now present in this SDF file. Okay, let me show you in a text editor. You can either open it in TextPad, or you can use another open source software like Notepad++ will also do. This is also a free software. You can use this as well. You can download it from internet. Okay, and you can view the same thing in Notepad++ also. So it is not necessary that you should possess TextPad, otherwise things won't work. You can also use this any text editor. Okay, so this Notepad++ is free and online, right? You can obtain it easily, right? So as we have already discussed, the structure of a typical flat file, or you can say the structure of a typical SDF file, it begins with the name of the molecule, which we can say that this is 2 prime hydroxyflavone, right? And it ends with a $4 sign, right? So this is the first pose starting from here till here. The second pose starts from the next line and ends with another dollar sign that is still here. And you can see that the binding free energies are also recorded in these SDF files. Okay, and one more thing, important thing is that the pose with lowest binding free energy are already present on the top, right? So this is the first pose which has been saved over here. It is usually the one which is having the lowest binding free energy, and that is what we are interested in, right? So all the 10 poses in this way are saved, or you can say exported in the SDF file. That is what we have done. Okay, so this is just for showing you that things can also be done in Notepad++, right? But I am comfortable using that TextPad. So next time onwards I would be using this TextPad only. Okay, so in this way you have created poses, right? For I have shown you how do you extract the poses from docking log file into SDF file. That is what we have done. I would again show you, do the same thing for our next pose or for our next molecule. Okay, now what we need to do is that we need to delete everything, right? We have deleted the data from docking log file. In tables the data also appear in docking results. I would also delete that, and from molecules I would also delete this, remove from the scene, so that everything is as we have begun. Okay, so again I would go to this loading the docking log file. Okay, go to the folder where you have saved all the docking log file. This time we would use our second molecule, 4' methoxyflavone. We have already done with 2' methoxyflavone. What is the second one? 4' methoxyflavone. Open it. Of course you would see that there are again 10 poses that are present over here. Okay, now we would kind of extract, or you can say export, all of these poses from docking log file into a separate SDF file by using this button, save as SDF. Go to the folder where you are supposed to save all the poses, right? And give a name over here. This is what? This is 4' methoxy flavone, and of course we are going to save all poses. Therefore all poses dot SDF would be appended to it. And then when you save it, you would see that after saving it another molecule would appear over here, right? Save, right? And if you go to the poses, now you see there are two molecules. Initially there was a single molecule, right? So in this way you would extract the poses for all the DLG files that you have saved. I have shown you how do you do with 2. The remaining would be your assignment. Okay, to extract all the poses in SDF format and name them as all poses. Okay, but now we won't be interested in all of these 10 poses, right? Because the binding free energy is different for each and every pose, right? And we would be interested in that particular pose who is having the lowest binding free energy. So in this case you can compare the binding free energy and the poses over here. The first pose has got the binding free energy of minus 8.3. Second pose, third pose and fourth pose, fifth pose as well, they have got the same binding free energy, minus 7.99, and the subsequent poses have got something else value, some values. Okay, so what we are not interested in all 10 poses. We are interested in a single pose with the lowest binding free energy. Okay, so now what we will do, of these 10 poses that are present over here, you can open it up in a text pad or any your favorite text editor, and we would separate the best pose, which I already told you, which is saved in the very first place or in very first position, right? We would copy the very first pose, the data for first pose, go to edit, use this copy button or simply push Ctrl C to copy. Okay, and then you can use this file new option. Okay, a new document is created, go to edit and then paste it. So in this way, from these 10 poses you have copied the data of the best pose, which is having the binding free energy of minus 7.85, right? Okay, so once you have done it, we would save it as an SDF file, right? To save it, go to file, then save as. Okay, and you can save it as 2 prime hydroxyflavone, but we have best pose now, right? We would name it as best pose.sdf. So in this way you can distinguish what SDF file contain all poses and what SDF file contains the best pose out of 10. Okay, and we would save it. Okay, maybe I will close this text pad. Now you can see that there are two files that save data on 2 prime hydroxyflavone. Okay, the very first file that has got name all poses will have all of the 10 poses saved in a single SDF file, from which we have extracted the best pose in a separate file. Okay, this file contains a single pose, this file contains all poses, and it is also reflected in their size. Okay, the file which has got 10 poses is bigger and the size of the file which has got a single pose is smaller. It is only 2 KB, right? We would do the same for our next molecule, 4 prime methoxyflavone. Open it in textpad. The data of all the 10 poses is present over here. What are we supposed to do is to separate the data for the best pose. We have copied it, go to file, new, right click it and paste it. Okay, check out the binding free energy is minus 8.3. You have got the best binding free energy pose is minus 8.3. So we would save this pose separately. Okay, so in this way you have separated the data, then go to file, then go to save as, right? And use, and you can rename it 4 prime methoxyflavone, but instead of all poses we would rename it as best pose, bestpose.sdf. Make sure that you have this SDF written over here, otherwise it would be saved as a text file, right? Okay, then hit this save button. Now you can see that there are two files for 4 prime methoxyflavone, the one that contains all poses, another one that contains the best pose. Okay, so in this way let us revise what we have done. We have started with loading the docking log file in our Python prescription, and from Python prescription we have extracted all the 10 poses that were present in that docking log file. In a separate file we have named it as all poses.sdf, right? And from this all poses we are interested in that one pose which is having the lowest binding free energy value, and we have separated it into a separate SDF file. So this file would contain your best pose but in SDF format, right? So now we have the best pose in SDF format, right? And we also have the structure of our protein molecule, right, which we have used, right? apo3nt1a. We can copy this file, right? How do we obtain the complex, right? And we can create a new folder in which I will give the name as 3. In this folder we would generate all the complexes, right? And for generating complex we would require our protein molecule as well, right? So this is where it is. So in this way, now your assignment is that whatever all poses that you might have generated for all the ligand, you would have to separate the best pose out of them, right? So in our next step, uh, we would need this SDF file which contains your best pose, right? But there appears to be one problem. In order to generate complex, your target molecule is still in PDB format and your best pose is in SDF format, right? So you cannot merge a PDB file with an SDF file. So what we can do alternatively now is that we would have to convert this SDF file into PDB file and then use that PDB file to generate a complex with your protein molecule. Okay, so that is why we need to generate, or we need to convert this best pose which is currently in SDF format into PDB format. Why? Because we want to generate the complex and your target protein is in PDB format. Okay, so in order to merge your ligand and your receptor, they should be present in the same format. Okay, now how do we do this thing now? This is really very interesting. So our new objective now is to convert this best pose which is present in SDF format into PDB format. Okay, and this format conversion is usually done using software called as OpenBabel. So I have already installed this OpenBabel GUI, right? I would ignore this now and I would use the data that I browse from the file, right? So now you can see as to in what format is our best pose. It is in this SDF format. So our input format over here would be, we have to change the input format over here from smiles to SDF, right? Then I would load our SDF file. Okay, we are still in this compound library preparation. Move your folder, right? Move your folder to desktop, then go to docking lecture and obtaining complexes. Where are your poses now? We are still in this poses folder and our best pose is in this SDF file. Therefore select it and open it. Now you can see that the data for 2' hydroxyflavone is present over here, right? And we want to convert this SDF file into PDB file. Therefore you would have to select over here what is the output format. We want to convert it into PDB file or protein data bank format. Okay, and we would have to first create a file in which all the data would be saved. So we need to create this PDB file first. So in order to do that, go to this browse button. Okay, and in the same folder poses, you might want to create a file called as 2' hydroxyflavone best pose. Instead of SDF, now you would give extension as PDB because that is the file format that we want to save our file, right? So make sure that you rename it to PDB over here. Okay, and then hit this save button. This would create a file name 2' hydroxyflavone best pose.pdb, and all you have to do is to hit this convert button so that your SDF file would be converted into PDB file. By hitting this convert button, this SDF format has been converted into PDB format and it would show you a message over here that one molecule is converted. That is what we need to do. Okay, right? So we have converted. Now you can check out in your poses folder that your molecule base pose now is in SDF format. We would do the same conversion for in case of this 4' methoxyflavone, right? So let me do it. So you have already selected, this is already in SDF format, right? All you have to do is to browse. Which is your next pose? Yes, this one is the one. This is the file 4' methoxyflavone best pose. And when you hit open, you would notice that the coordinate will change and the number of lines would change. Okay, hit open. Okay, and you can see that the data has changed over here, right? The output is still in PDB format. You don't need to change it now. Go to this file, create a new file wherein this SDF data would be saved in PDB format. You would name it as 4' methoxyflavone base pose. Instead of SDF, you would need to change it in PDB and then save this file. Okay, and use this convert button, and it would say the molecule has been converted. Again check, this new molecule has been appeared. Okay, so now we have got the best pose in PDB format. We would copy these best poses now, this and this. I would right click, copy it. Okay, maybe create a new folder called as complexes and paste it over here. So now for creating complex, we have everything. We have a protein molecule which is in PDB format now, and we have the best poses that we have predicted in our docking software. They are also in PDB format. Okay, all we need to do now is to create those complexes. Okay, so now creating these complexes is also a very easy job. What you can do is that all you have to do is to right click it, open it in text editor. Okay, it would open it in a text editor. Go down. Okay, this end mark indicate that the information of file is ended. Okay, so what you can do is that you can go to the previous line, hit an enter, give a termination mark over here. That would indicate, this ter line would indicate that information of protein ends over here, and whatever is present after this termination line would belong to your ligand molecule. Okay, so next thing you need to do is to open your ligand file in textpad. Open it up in textpad. This is your ligand molecule. Copy all the information that begins with heteroatom line as well as connectivity line and the master line, right? Copy it and paste it after the termination line. Go to edit, then paste. Now you see that the protein information is till this line, then termination line indicate that the protein information end here, and from the subsequent line you have information for your ligand molecule. Okay, so in this way you have created a protein ligand complex. Okay, now what you have to do is that you would have to file and then use save as. Okay, so you can give it a name. You can give it a name like COX2. This is 2' hydroxy flavone complex. Since it is an all file written over here, you would have to specifically mention the extension PDB. This is supposed to be a PDB file, right? And now let us save it. Right, you can close it now. So now do you see over here, this complex file has appeared. Right, now let us try to visualize how our complex might look like in SPDBB. Open the SPDBB. Here is your hard earned complex molecule that you have recently generated. Just drag and drop it. Here we go. Here is your protein molecule, which you can show it by the ribbon representation. Scroll very down, and here is your flavonoid molecule that you have docked. And now you can see that this flavonoid molecule is docked exactly where your ligand molecule or your naproxen molecule was. Right. So in this way, you generate the protein ligand complexes. Right. So let me do the same for your next molecule. Let me close this. Let us begin with generating the protein ligand complex for your second molecule, that is 4'-methoxyflavone. How have we done it? Open that APO3NT1 chain A, open it up in TextPad, scroll down, go to the previous line, hit an enter, place the termination mark over here, hit an enter, and maybe you can save it so that every time you don't have to do this. Right, now you have already saved it. Right, so next time you open it into TextPad, just scroll down and this thing would be already there. So this is your receptor molecule. All we have to do is to open your best pose that you have converted into PDB file in text editor. Copy all the lines that begin with heteroatom, connectivity and master. Okay, right click, copy it, go to your target molecule, right click, paste it. Okay, and don't hit save button. Okay, otherwise this file would be overwritten. This protein file would be overwritten. Right, you have to do what? Go to file and save as a separate file. If you hit the save button, this file would be ligand information would be permanently saved over there. So you would save it as a separate complex. Right, then again, of course, you can save it in a different name. Let us COX2 would use the same name, right? But instead what we have got 4' methoxyflavone complex .pdb and save it. That's it. Again you have generated another complex. Well, let us again visualize it. You can visualize it in SPDBB. Right, 4' methoxyflavone. Let us drag and drop it over here so that you can see it. Okay, again show your protein molecule in cartoon representation, ribbon representation, and here goes your ligand molecule in your protein. This is your protein complex. So your assignment now would be to generate all of such protein ligand complexes. Okay. Okay, so in this way you generate all the complexes. Now let us start uh, performing the interaction analysis. Interaction analysis can be done. Why do we do such interaction analysis? Interaction analysis is done to test if your new ligand molecule forms hydrogen bonding interaction with the neighboring amino acid residues or not. right? If it forms van der Waal contact, or if it forms hydrophobic contact with the neighboring amino acid residue. If you want to know those things, you would have to perform interaction analysis, right? But to perform interaction analysis, you must have the complex of your protein ligand molecule first, and that is what we were dealing with for time being. Okay, so you can perform interaction analysis in two ways, right? You can do it using the traditional way that I have taught you. Open your SPDBV, right, and load your protein ligand complex. Okay, and how can you perform, or how can you check if the ligand molecule forms hydrogen bond or not? We would have to focus only on ligand molecule, select it, hit enter, and then maybe you can go to select, and then you can select neighbors of selected residue. We would select the groups that are present in vicinity of 3.5 Armstrong, so that we would select only groups that are present in the vicinity of 3.5 Armstrong. Okay, and then you can use the show button to show whatever the residues that are present in 3.5 Armstrong vicinity. Okay, then we can also label them, use this zoom center button, right? So now you can see that there are a lot of amino acid residues that are present in 3.5 Armstrong vicinity. Now let us see if they can form hydrogen bond, right? For that you can go to this tools and then compute hydrogen bond. Yes, now you can see that this serine 530 forms the hydrogen bond with the oxo group present in your flavonoid, and this residue Arginine 120 and tyrosine 350 might form the hydrogen bond with the methoxy group present over, OCH3 group present over here, right? So in this way you have located at least 3 hydrogen bonds that our ligand can form, right? So this is one of the way to detect if your new ligand, how your new ligand might stabilize the protein ligand complex. Okay, by manually doing it, by manually checking if it forms the hydrogen bonds with the neighboring amino acid residue. This is one of the way. Or there is another way by which you can document these interactions as well. Okay, and that can be done using another software called as LigPlot Plus. I will just show you. So this is one of the way to identify if your protein ligand complex would be stabilized by hydrogen bonds or not. Okay, or how many hydrogen bond it can potentially form with the neighboring amino acid residues in from your target protein. Okay, so this is one of the way. You can close it. Another way around, we don't need this open label, is using a software called as LigPlot Plus. So this LigPlot Plus can be used to visualize your three-dimensional active site into two-dimensional plots, what we call it as LigPlot. Okay, so what you have to do is that you have to open this LigPlot software, go to file, open your PDB file, go to this browse button. Okay, again locate where your PDB file is located, where your complex is located, right? It is located in this lecture, obtaining the complexes, then you can open this 4' methoxy flavor, any of the complex, and open it. Okay, now it would ask you that if there are multiple ligand, which is the ligand you want to focus in your 2D plot. So since we have got a single ligand over here, we would select the name of our ligand and hit this run button. Look, it has detected and it has converted into a two-dimensional plot, what we call it as LigPlot. Okay, and over here you can see that this oxo group that we have detected is forming the hydrogen bond with serine 530, and the oxygen from methoxy group is forming two hydrogen bonds with tyrosine 355 side chain and arginine 120 side chain. Okay, so in this way what you can do now, and one more thing, using this LigPlot you can also detect as to what are the amino acid residues that might form hydrophobic interaction with the hydrophobic portion of your ligand. Okay, so this valine 116, alanine 527, glycine 256, tryptophan 387, all these amino acid residue are in hydrophobic interaction with the hydrophobic rings that are present in your ligand, as you can see over here. Okay, so this is how you have converted your three-dimensional active site into two-dimensional plot. Okay, so now we would want to save this plot into a PDF file. For that, you can go to File and use this print screen option. Okay, and I have already installed a software called as Do PDF. You see this? You have to install this software called as Do PDF. It is also one of the free and open access software that if you install, whenever you print something, that would be converted into PDF file. Okay, by using this Do PDF as a printer, and if I hit OK, you would be saving this particular view in a PDF file. You can go to Browse and tell where this LigPlot is supposed to be saved. Go to Desktop. Okay, go to Docking Lecture, Obtaining Complex, and maybe we would create a new folder and name it as 4 LigPlot. Okay, and this LigPlot belong to which belong to what? To the complex. What was the name of that complex? It was 4 prime methoxy flavor, right? And just hit this save button and hit OK. Okay, so by doing this, in the newly formed LigPlot folder there is a file with 4 prime methoxyflavone, and in this way you have saved this view from this LigPlot software into a separate PDF file. And over here you can also try to identify that this hydrogen bond has hydrogen bonding distance of 3.18 angstrom, while this hydrogen bond is more stronger. Why? Because the distance is short. It is this hydrogen bond is of 2.47 angstrom. Okay, so in this way you can save your results, and then maybe you might want to include these results in your research paper, or maybe you want to include these results in your project report, or whatever way you want to express it. Okay, so this is how you do it. This is how you create a LigPlot. I will again show you how do we do this for your next molecule. I would no, I don't want to save it. I have already saved it in PDF file. Then go to again File, open a PDB file, go to Browse, right? And let us create a LigPlot for this 2 prime hydroxyflavone. Again hit open, select what ligand do you want to focus on and hit run. Okay, so here is how your 3' hydroxyflavone might interact with your COX molecule. It will form hydrogen bond with arginine 120 and tyrosine 355 through the substituent hydroxyl group that is shown over here, right? And again we would save it as a PDF file by go to file, use this print screen option, change the default folder to do PDF, and then hit OK. Give some name, browse to the file. What is the name of this complex? 3' hydroxy flavor. Click it on save, click it on OK, and within a second of time your view has been saved in a PDF file, and you can open this PDF file anytime later on. When you are done with this, you can close this thing. Do you want to save the change to the current file? No, everything is okay. So now your job would be to convert uh, to create this LigPlot for all the remaining compounds. So in this way you perform the interaction analysis. Performing interaction analysis means what? To figure out as to how your ligand might be stabilized in your protein molecule, right? To know as to what are the possible polar groups that are present in my ligand molecule that can form putative interaction with the side chain or the residues that are present in its vicinity. Okay, so in this way you can check it out, how many hydrogen bonds are there, how many residues form hydrophobic interaction, so on and so forth. Okay, so this is how you do it. This is how you perform. This is how you create complexes of your protein ligand structure and perform interaction analysis. So in our next lecture I would show you as to how do you create a publication quality images using PyMol. So see you guys in our next tutorial.